This video will be helpful to preclinical researchers who are particularly looking for alternatives to animal tests, various laws and regulations related to animals, and what we can do in order to avoid or replace the animals used in preclinical research. So, where or what are the areas where laboratory animals are mostly used? So there are three main areas where laboratory animals like rat, mice, guinea pig, rabbit, these animals are used. First one is for biomedical research, second one is product safety testing, or this can be drugs can be tested, cosmetics, household cleaning products, pesticides, industrial chemicals, etc. This safety assessment of these things can be done using animals. Third one is for education purpose, in order to make students understand about research or to give demonstration. So we will see where, particularly in pharma industry, in drug discovery and development phase, where these animals are used. So these animals are used in preclinical stage, where the drugs are tested in animals. Now we will see what are the scientific reasons for why moving towards alternatives to animal experiments. So in the left pie chart, we will see percentage of animals used, that is mice, rat, fish, birds. These are used in different procedures. So around 72% of mice are used in laboratory procedures for various purposes. So what are scientific reasons why we should move towards or we should find alternatives to animals? These are because of three main concerns, that is scientific, economic and ethical concerns. What scientific concerns are? Scientific concerns arises because of extrapolation of data from animals to humans. Also these procedures and animal experiments are questionable, and this how they are relevant, because we know we are ultimately going to give our drugs to humans. So how testing in animals would be relevant for our purposes. And reproducibility, it means can we get the same results after doing repeated experiments on the different animal species. Second reason is economics, because these animal procedures are very costly, labor intensive and time consuming. So it also important to know economic aspect of using animals. Third one is ethical concerns, because animals are exposed to different pain inducers, or it causes some drugs or toxicant causes various distress, and societal concern means people are also aware of not to use animals. Supposedly we see in India how people are concerned about cows and even for the small animals. So that's why you must have heard of the PETA, that is People for Ethical Treatment of Animals. This is non-profit organization and its slogan is animals are not ours to eat, wear, experiment on, use for the entertainment or abuse in any other way. So PETA is working, or it works mainly for protection of animals which are used in various unethical purposes or for the entertainment or anything people use for. So it has four core issues, that it opposition to factory farming, fur farming, animal testing and the use of animals in entertainment. So this the next thing is AALAC. So it has nothing to do with the PETA, but I have just put it for your information that what AALAC is, that is Association for Assessment and Accreditation of Laboratory Animal Care. Because of more, if you are using animals, but if you use it under AALAC guidelines or AALAC accreditation, then it can be considered that you are following proper procedures, so that the animals will not suffer more, or if they are suffering, then how preventing or precautionary measures you are taking. So, what are alternatives to animal testing? Development and implementation of test methods that avoid the use of live animals. So there are two major alternatives: in vitro techniques and in silico computer simulation. So there are also various types of alternatives to animal experiments like cell culture and tissue engineering. So skin corrosion and skin irritation models like epiderm, episkin and skin ethic reconstituted human epidermal models are available. Then computer simulation, which includes computer operated manicures, which are also known as crash test dummies. Then for medical imaging, how drugs are metabolized that can be tested by using microdosing in humans. So this is also one of the alternatives to animal experiments. Then microfluidic chip, then fungal model for mammalian drug metabolism. We'll see in brief of each one of these in upcoming slides. So in vitro techniques are studies which are performed with microorganisms, cells or biological molecules. So which are also called as test tube experiments because they are conducted in very short microenvironment. So what are the advantage of in vitro techniques? That it reduces use of animals, it gives large scale production, decreases the need for experienced laboratory personnel because suppose you need to inject to the rat, then you need to have proper knowledge of how to inject, by which route you have to inject, and so many other things. Then next advantage is species specific, simpler, more convenient and more detailed analysis. Next advantage is replacing studies in whole animals. Then next is simplicity, and next is convenience and automation. So you can do some of the techniques by automatic, but you need to perform everything manually in animal experiments. So what are the disadvantage of in vitro techniques? That in vitro to in vivo extrapolation. So if you got the results, but how these results you can extrapolate to in vivo, so that is one of the concern. Second is xenobiotic metabolism. Third is extrapolating from biomarkers in vitro to adverse effects in vivo. And fourth is interaction between different cell types. So here I have mentioned one example of CACO2 model, which is in vitro technique. Then in this model is used particularly for studying permeability of compounds in order to evaluate mechanism of transport and for predicting the in vivo absorption of drugs. So these are cells CACO2 are cells of heterogeneous human epithelial colorectal adenocarcinoma, and this is how you can see in the figure, this is how these CACO2 cell monolayer have been placed, and one side will be the apical side where your donor compartment, and then basolateral side, and this structural mechanism we'll see in the upcoming videos if you want to get the information about this in detail. So how it measures, or what it measures? It measures the rate of transport of compound across CACO2 cell line, and its analysis can be done by LC-MS. So we'll see in this 21st century toxicity testing, which 3D tissue models are available. So there are skin corrosion and toxicity guidelines are there, particularly accepted by OECD, that is OECD 431. For skin irritation, toxicity guideline 439. For eye corrosion and irritation, toxicity guideline 492. So epiderm, episkin and skin ethic reconstituted human epidermal epithelia model are derived from human skin cells which have been cultured to produce a model of human skin. So they are derived from human skin. So how they are, we need to culture them for the particular model, that we will see in the upcoming slides. So these two agencies we will see in short how they are concerned with the alternatives to animal experiments. So one is European Union Reference Laboratory for Alternatives to Animal Testing, and second one is interagency coordinating committee on validation of alternative methods. What it does is it scientifically and regulatory acceptance of non-animal tests. If you have any idea of developing any model, then you can take help of them, so they can give you a brief on how to, or help you in designing new animal, replacing in place of animal. You can go for new alternatives to animal test. So what are advantages of 3D models? That metabolism is one of the advantage, because you can add xenobiotic activators or deactivators, or in other words, if I can say, you can add enzyme inducers or inhibitors into it, so that your metabolism you can control. Then next is diversity of measurable parameters like histology, enzymology, biochemistry, imaging, transcriptomics, and many more. Third is barrier function. So you can you will have particular model for checking barrier function like topical or systemic application, which mimics the real time conditions for use and exposure, like skin models. So here is the procedure of how you can reconstitute human epidermal cell model. So here I have shown particularly epidermis reconstitution. So you need to take skin by using skin biopsy, then you need to extract an amplified keratinocytes, then seed these keratinocytes on a support which is made up of collagen or polycarbonate. Then the proliferation of keratocytes in immersed conditions by providing suitable environment. And at last, before going to the actual experiment, please air expose them to induce stratification and proliferation. This is how these cells are reconstituted. So what measures you have to be taken before developing or while going for the alternative models? So there are some of the things you need to consider, like certificate of analysis, standardization, quantity, trend traceability, quality control, reproducibility, stability, biological safety controls, and many more. So here is one example of in silico techniques, that is computer simulation. So what in silico means in Latin term, it is used for in silicon, which means the mass use of silicon for semiconductor computer chips is an expression used to mean performed on computer or via computer simulation. So there are various in silico models like quantitative structural activity relationship, then crash test dummies, then human patient simulators. So we will see them all one by one. Here is one example of trauma man, because this mimics the accidental type of conditions or it simulates the accidental type of conditions. This is just a dummy of some accidental exposure kind of thing, so which replicates breathing, bleeding, so everything will be there like a human, but which is injured, or how what will happen if it gets injured by suppose accident with a car or supposingly hemorrhage or anything. So it gives realistic layers of skin and tissue and ribs and internal organs. So what are mathematical and computer models? That is, one is QSAR, that is mathematical models of quantitative structural activity relationship. These models are used to predict biological responses on the basis of physical and chemical properties, structure, and available toxicological data. The limitations of such models are due in part to a lack of understanding of the mechanisms by which toxic effects occur, because we need to ultimately understand, okay, it is fine that toxicity is happening, but we need to cure it. But before curing, we should know how it is causing the toxicity, then only we can take best measures in order to tackle that issue. But by using these models, we cannot understand by which mechanism it is causing toxicity. So next is the biological effects of chemicals are expressed in quantitative terms. Another approach more broadly applicable is to examine the contributions of various portions of a molecule. Next is quantitative structural active relationship properties. So what properties you need to consider if for the compound, that is hydrophobic parameters, because more hydrophobic is the drug, it will get penetrate more, and then we will be responsible either for toxicity or pharmacologic effects. So we need to understand this all parameters, that is aqueous solubility, octanol water partition coefficient, then total surface area and total molecular volume, and many more. So one of the alternatives to animal experiments is to use humans in place of animals. For example, microdosing, which is non-invasive technique, non-invasive imaging technique actually, which so many kinds of research can be done by using these techniques, like dietary research, pain research, and human cell and tissue culture can be also done. So microdosing is for measuring how very small doses of potential new medicines move around the body, because some drugs we have to take in a very small quantity, but where it is getting metabolized, or so we need to have particular target, because we have very small amount of drug and which is very potent. So we want that drug to act to the particular side, because we can't give much drug, so it should not get metabolized. So we need to identify where it is getting metabolized or what excreted. So we need to identify it by using imaging. So that's why this technique is very used, and these microdoses are radio labeled so that we can see it in the outside environment. Then volunteers to measure how very small doses of potential new drugs behave in a human body. So what are the disadvantages of this method? That we cannot predict the toxicity or side effects that occur at higher therapeutic doses. So we are sure about this, but we don't know what will happen in the large doses, because we don't test it for the large doses for longer period of time. Next available alternative model to alternatives to animal testing is microfluidic chip. So the choice of material used for design of this chip is PDMS, that is polydimethylsiloxane. The size of these chips is very nearly about 2 centimeter wide, so that is why it's called as microfluidic chip. That is not the exact cause actually, because here we'll see the very small amount of small organs are placed on the chips, and then these are connected like in human body. These organs or everything is connected or supplied with the blood. So this is how these things are created in very small chip. That's why it's called as microfluidic chip, because of very little amount of fluid is available in the series of small chambers that are engraved into this chip. So, and the chamber composition, finally it would be like what I said, is a sample of tissue from different parts of the body can be placed, and then its substitute of blood is through microchannels, which are shown in the figure, and the analysis of this can be done by computer by using sensors which are placed in the chip. So what are the applications of this thing? So discovering mechanisms of human disease and pathway of toxicity, identifying human responses to drugs, toxins, then testing drugs and chemicals, then modeling pharmacokinetic and pharmacodynamic properties of drugs and chemicals. So these things, microfluidic chip is very potential to be used in various pharmacological or toxicological assessment. So this is how, if we connect each organs, and like that we will have whole human organs on a very small chip. So that is why it is called as human on chip in a fancy way. So Drosophila is also one of the alternatives to animals, although it is if we consider it as an animal, but because of its certain characteristics we can use in place of like rat and rabbit and other big animals. We'll see this how why Drosophila, because it is most extensively used and most well understood model. Then also it measures approximate its length is approximately 3 millimeter, so it takes or requires very small amount of space to live in. So one female may lay 30 to 50 eggs per day throughout her lifetime, which is if we consider for rat, it litter size of rat is around 10 to 12, so it is more convenient. Then Drosophila are ideal for study the genetics and development. Then the complete genome sequence of Drosophila was published in 2000. So I will discuss in detail about genetics and development. If you want to study genetics and development studies in rats, so you need to go for the first generation, second generation studies, where you need to have their pups to be grow bigger size, and then their second generation will come. Then it is lengthy procedure and it requires more number of animals. Next, why we should use Drosophila is the relationship between fruit fly and human genes is so close, that is about 75% of the genes that cause disease in humans are also found in the fruit fly. Okay, so also Drosophila have a short simple reproduction cycle. It is normally about 8 to 14 days only. Fruit flies are very small and but so small that can't be seen. Okay, so next thing is fruit flies are very small, but not so small that they can't be seen without a microscope. They are inexpensive to maintain in the laboratory, as it requires less space. Also they require simple diet consisting of simple source of carbohydrate, which is of corn meal, and proteins, which is of yeast extract. Also other sources are considered to be alternatives to animal experiments, which are information resources including literature, website, then images and video presentations, and also background case studies. What are the limitations of alternative models? That reliability, subjective assessment, variability, differences in anatomy and physiology. These are some of the limitations or major limitations of alternatives to animal models. So what you can do to develop and validate alternative methods? Funding for biomedical research should be redirected into the use of epidemiological, clinical, in vitro and computer modeling studies instead of cruel experiments on animals. Good animal welfare you should maintain, and also government, industry and societies to work collectively. So this is one of the quote. The difficulty lies not so much in developing new ideas as in escaping from old ones. So like हिंदी में ऐसे बोलते ना कि आप ऐसा नहीं कि नई चीज़ आप जल्दी से इनकॉर्पोरेट नहीं कर सकते, बट पुरानी चीज़ आपको पुरानी जितना टाइम या जितनी मेहनत आपको या डिफ़िकल्टी फेस होगी पुरानी चीज़ें छोड़ने में. सो ह्यूमन का ये नॉर्मल बिहेवियर है कि वो जल्दी से पुरानी आदतें नहीं छोड़ता है. So like that. So more science, less animal used. Thank you for the attention. Thanks.